in when that quiz is given. There's a mentoring session Thursday night in this room. Probably wouldn't be a bad idea if you need to do the retake to stop by the mentoring session. Austin can go over the questions with you and explain why the answer that you put for a particular question was wrong, if indeed it was wrong. It won't be the exact same quiz. There will be variations, okay? But I do encourage you to go back to your old quiz and make sure you understand what the correct answers are, because some of the questions may be repeats. It will also, again, be 20 multiple choice questions. Same kind of thing, just a little bit different answers, but still addressing the same major concepts. Okay? Any question about that? Anything about lab, Dr. Morton, that needs to be...
Just be on time, please.
Be on time?
Actually, be a little early.
Okay, let's start out with this. One of the questions that I like to ask on an exam, and you'll see these on old exams that are posted on the website, are agree and disagree questions. Agree and disagree questions require you to do some thought and some justification, because typically these questions are worded in such a way that sometimes they're open to interpretation. And so it is not uncommon for me to give full credit for someone who has agreed with the statement, and also give full credit to someone who has disagreed with the statement. It depends upon how you interpret certain phrases and then how you justify things. When you do these, don't just put agree or disagree. You get the majority of your points by explaining what your logic is. Now, this is something that you may or may not be able to answer from last semester. But there's several things in here that maybe we should spend a little bit of time unpacking. And this also is going to kind of relate to some concepts that we're going to start talking about as we introduce our tour of the cell. So the statement says, phylogenetically, an amoeba and a human are more closely related than an E. coli and a halophilic bacteria living in the Dead Sea. Okay? Just out of curiosity, how many of you would agree with this question, this statement? How many of you would disagree? And how many of you have no clue? Okay, what's the problem? There might be some terms here that you don't understand. Okay? When you look at these agree and disagree questions, typically there are some key vocabulary terms in there. If you really struggle with agree and disagree questions and you're not quite sure how to approach them, one of the things you can start doing is start kind of define terms that are in the statement. Tell me something. Okay, don't just leave it blank if you're kind of stumped. Okay? What is phylogenetically referred to? Does anybody know? Okay, classes and orders and families. It really has to do with evolutionary relationships. So what this statement is saying is, evolutionarily, an amoeba is closer to a human than an E. coli is to a halophilic bacteria. What else is stumping you in that particular statement you might want clarification on, besides phylogen? Halophilic. Does anybody know what that means? You know anything about the Dead Sea?
Very salty.
It's very salty. And actually there are bacteria that can tolerate very high concentrations of salinity, such as you find in the Great Salt Lake or the Dead Sea. And they're called halophilic bacteria. What about E. coli? What do you know about E. coli? You've heard of it before, right?
Is it found in like meats and stuff?
It can be found in meats and contaminated meat. Where else can it be found? It can be found in water, particularly if water is polluted.
Stomach digestive system.
Not in your stomach, but in your intestines. Okay, it's a gut bacteria. E. coli is one of the more well-known, well-studied bacteria. It's sometimes referred to as kind of the lab rats of the microbial world. We know a lot about E. coli. Okay, so this statement basically says, if you tear it apart, that these are in a more similar grouping of hierarchies than these are. Okay? What is the largest or most inclusive group of hierarchies that we assign organisms into to try to start determining what their evolutionary relationship is. Remember your hierarchies and your groups? How do they go? What's the first one? What's the most important? Kingdom, phylum, class, order, family, genus, species. That is a traditional hierarchy. That's what Dr. Morden and I grew up learning. It was proposed by a guy named Whitaker. How many kingdoms are there? Anybody know? Does anyone know what any of the kingdoms are? What are we? What kingdom are we in? Animalia, animal. It's another kingdom. Plants. There are fungi. There are protista, which are a lot of single-celled organisms that some of which we'll be looking at in lab next week. Is that right? Okay. You may know what the last kingdom is. Animal, plant, fungi, protista. Now we're going to get a little, it's called prokarya. Okay. Does anybody know how prokarya kingdom differs from the other four kingdoms? Prokarya. Has to do with cell type. What are the two major fundamental different types of cells? Prokaryotic and eukaryotic. Okay? How do they differ? Prokaryotic cells don't have membrane-bound organelles, including a nucleus. Eukaryotic cells do. So plants, animals, fungi, protista are eukaryotes. The bacteria are prokaryotes. Okay? Under the old five-kingdom approach, E. coli and the halophilic bacteria are in the same kingdom, prokaryote. Okay? Amoeba and humans, depends on who you talk to. Okay, some people will put amoeba in the protista, others will put it in the animalia. So right there, depending upon your assumption of how things are classified, you might answer that differently. If you're a person that believes there's evidence for amoeba and humans to both be in the same kingdom, animalia, then you would say, no, they're no more evolutionarily different than the bacteria are. These particular bacteria, these halophilic bacteria, are categorized into a group of bacteria called the archaebacteria. So you know what archae refers to? The prefix arch means? What is archaeology? Ologies are studies of, right? What's archaeology?
Past.
Study of old things. Okay. The archaebacteria are referred to as the ancient bacteria. We think that these are some of the very first bacteria that evolved. And many of these archaebacteria live in very extreme or hostile environments, like boiling hot springs or very salty environments. And they're called extremophils. They love, phil means to love, extreme environments. Well, probably about 15, 20 years ago, a molecular biologist named Carl Woese started looking at this hierarchy, the five kingdom approach, and said, does this really reflect reality? And does it really illustrate evolutionary relationships the way it should? He was a molecular biologist and spent a lot of time sequencing, determining the order of A's, U's, G's, and C's in various ribosomal RNA molecules. And he started looking at all this data and he said, you know, I think the five-kingdom approach to classification is outdated and doesn't reflect reality of how things are really related evolutionarily. So he suggested the addition of a new category above that of kingdom. Does anybody know what we call those? Domain. Okay? So there's domains, and then there's kingdom, and then phylum, etc. Okay? Anybody know how many domains he suggested? 3. Okay? And the 3 domains are the Archaebacteria, the Eubacteria, U means true, and the Eukarya. So what had in the five-kingdom approach previously been members of the same kingdom, the kingdom Prokarya, he split out into 2 separate domains. And then everything else, everything that was eukaryotic, was in the 3rd domain. That was a revolutionary way of looking at things, but his ribosomal RNA analysis suggested that evolutionarily, these were much different and had a different evolutionary history than these were. So basically, he took one kingdom and split it into 2 domains, and then the other 4 kingdoms were placed here. So if you accept Wosey's 3-domain system of classification, then you would want to agree with that particular statement. Because both amoeba and humans are in this domain. The halophilic bacteria are in a separate domain, and the E. coli are in a separate domain yet. So because those amoeba and humans are in the same group, they're more closely related than things that are in different groups. Okay? This illustrates a couple of important things. One is that concepts in biology and science are constantly changing and evolving. The five-kingdom approach to classification was a long-held concept that made sense until new information began to accumulate. And that new information, which was based upon a new technology, the ability to sequence RNA, started giving us new insights, and then people looked at that and said, well, based upon this new information, this system now makes more sense than the previous system. And so the common layperson's perception of science, that it's a set of unchanging facts, is really totally wrong. Science is constantly changing and evolving. One of the things that we will see when we look at cell membrane structure is that the idea of how a cell membrane was organized has undergone an evolution over time. And what's happened is one model has replaced the previous model as more data is accumulated. And what do we have to do if our model or hypothesis, models or hypothesis, don't fit our data? We have to revise our hypothesis, right? So the models keep changing as new information starts developing. This is just an outline. I like to put these in when I think about it in PowerPoint slides, just kind of give you an idea of what we're going to go over in a particular section. So this is kind of a way to organize your studying a little bit. So, what do these organisms all have in common? Yes? Okay, they're all alive. They all have genetic information. They all have a certain level of organization. And they're all made up of one or more cells. Okay? Here's some cells, some human cells. Where did all these cells come from? They just poof up here. They came from another cell, right? Those two statements are the basis of one of our most important ideas in biology called the cell theory. The cell theory states that all organisms are composed of one or more cells and that all cells come from other cells. Now that does beg an interesting question. What question does that beg? Where did the first cells come from? Okay. Same slide or same pictures, but a different question. Is there any insights you can get about cells in general by looking at these various images? What strikes you? Yeah.
You can kind of get a feel for their function, how they're structured, like the nerves are long, so you can tell that they kind of like send synapses and stuff.
Good, good. Okay, so there are different kinds of cells, and they have different structures, and those different structures are adapted to different functions. So this idea of the relationship of structure and function. Does anybody know who's given credit for the developments of the cell theory? We all know, for example, who was credited with the theory of evolution, right? Who is it? Darwin, or somebody could be creative and say Wallace. Have you ever heard of Wallace? Okay. Well, it's really hard to pin down exactly who is responsible for the formulation of the cell theory. There were several people that contributed to it. Does anybody have any idea of what kind of general time frame in which the cell theory was proposed? How about a century? 1800s, yeah. Okay. That's a relatively new theory. Why did it take so long for the cell theory to kind of develop and to gain traction? Technology, and what technology specifically? Microscope. And so the development of the cell theory is very closely tied to the development of a technology, the microscope. Does anybody know who's generally credited with kind of the invention of or use of the microscope? You'll get a lot of disagreement about this one from history of science people. If you talk about the father of microscopy, does a name pop out at you? Anton von Leeuwenhoek. Von Leeuwenhoek lived in the mid-1600s. He was a very interesting guy. He was actually a linen merchant, a cloth merchant. And he developed his first microscopes to examine the cloth for flaws. And his first microscopes, which were these little crude handheld devices, were really kind of a glorified magnifying lens. But von Leeuwenhoek was also very curious and looked at a lot of different things. And when he looked at some water samples, he noticed something very interesting. And that was that there were these little organisms present in the water, which were invisible to the naked eye, that he was one of the first people to see and detect. He called these little organisms animalcules. Little tiny animals. As it turns out, they were bacteria, maybe some small or some large protozoans. But this has blew people's mind. To think that there were these things that were invisible that were living in the water you drank, for example. And this discovery really sparked some very interesting investigations, particularly in investigations that related these little animalcules and human disease. So the germ theory of disease is dependent on this idea of being able to detect and know about the presence of these single-celled organisms. Another cell biologist that is pretty famous is Robert Hooke. Robert Hooke was also in the mid-1600s. And one of his more famous observations was looking at very thinly sliced specimens of cork from tree bark. And when he did that, he saw an arrangement that looked like this. And he noticed that the cork was made up of all of these little box-like structures, which looked like prison cells to him. And that was the origin of the term cell to describe these particular little structures that made up the bigger whole. Well, when you start talking about microscopy, one of the things that you have to do is you have to start adjusting your perception and your understanding of size of things. Okay? So we can run down a hierarchy here of decreasing size from meters to centimeters to millimeters to micrometers to nanometers. Okay? This gives you your relationship between all of those different types of metric size. But what we find is that most plant and animal cells fall within this general range of size of about 10 micrometers to 100 micrometers, or 10 microns to 100 microns. Another thing that you should notice from this is that bacterial cells, which are prokaryotic, tend to be smaller than plant and animal cells, which are eukaryotic. Then if we start looking at some of the organelles that can be present in eukaryotic cells, like mitochondria, they're pretty small. Something like a ribosome is really small. And then we get down in here into individual molecules, and then actually the atoms that make up those molecules. The ability to see small things has to do with the property that I think is referred to in the lab next week on microscopy. It's called resolution. Okay? And resolution basically is the ability to see detail in small things. And the human eye has a resolving power that is pretty good, but not good enough to detect most cells or most cell structures. So in order to detect these things and observe them and study them, we need something that can extend our senses, a technology, like a microscope. This shows you the resolution of a typical compound light microscope, which we will use in lab. And then this shows you the resolution of a newer type of instrument called an electron microscope, which really dramatically increases our ability to see things like viruses and small bacteria, and even some of the cell organelles that are part of the eukaryotic cells. There are even newer microscopes called scanning tunneling electron microscopes and atomic force microscopes that really get us down into even this small molecular level. So as we improve technology, new worlds are opened up to us, and we learn more about cell biology. For example, until the electron microscope was developed in the 1950s, we really didn't understand that there were two fundamentally different types of cells, prokaryotic and eukaryotic. But once we had this ability to see really small things, we could see that bacteria have some fundamental differences in the way they're organized compared to typical plant or animal cells. Your textbook spends a little bit of time talking about some types of microscopy. There are various types of microscopy. The one that we will be using pretty much exclusively is called bright field microscopy. This is an unstained specimen of human cheek cells. By adding some stains, you can differentially highlight some of the cell structures within the cell. So staining can be a nice addition to this bright field microscopy. You can see down here the difference between a stained and an unstained specimen. Here you can see the nucleus, for example. But there are new technologies that are just opening up our understanding of cell structure. There's fluorescence microscopy, phase contrast, Nomarsky or differential interference, confocal. And as these new technologies developed, it's just like opening a new window into the world of studying cells. For example, fluorescence microscopy has led us to the understanding that the cytoplasm of the cell is filled with this really elaborate network called the cytoskeletal system. When we were taking biology classes, they knew nothing virtually about the cytoskeleton. Nobody really knew it even existed. Why? Because the technology wasn't available to visualize it. Electron microscopy is very cool. The previous types of imagery that I was showing you all were forms of light microscopy. The energy we're using to view the substance is light energy. Your ability to see small things and discern detail is related to the wavelength of the energy you use to view the specimen. And the shorter the wavelength, the more detail you can see. Well, visible light has certain defined wavelengths. Does anybody know what the shortest wavelength of visible light is? There's a spectrum going from blue to red. What's shortest, blue or red? Blue is a shorter wavelength than red. Okay, so ideally, blue light is a little bit more desirable for viewing things. That's why sometimes microscopes will have a blue light filter in the base. So you're looking at it primarily with blue light. Electrons also have wavelengths, and they are much, much shorter than visible light. So electrons can give you incredibly more detail. There are two basic types of electron microscopes. There are scanning electron microscopes and there are transmission electron microscopes. Scanning electron microscopes are really good at looking at surfaces. And they give you almost kind of a 3D type of appearance. Here is a scanning electron microscope of a cell showing you the numerous cilia that are part of that cell surface. Transmission electron microscopy involves taking something you're looking at and cutting it into very thin slices and looking at a piece or a slice of that object. And so here is a longitudinal section of that cell. And again, you can see a crosswise cut, basically, of those cilia that are attached to the cell membrane of this cell right here. So this gives you a very different view. This gives you kind of an internal view. And when we start looking at transmission electron microscopy, which we'll see some images of later on, it just gives us this whole new insight into how a cell is organized. And we can actually see structural details of things like ribosomes and chloroplasts and mitochondria. You can barely see those with a light microscope, if at all. So it's a whole new window into understanding of cell structure. Okay, here's a type of image that you'd be expected to see in most textbooks. Is this cell prokaryotic or eukaryotic? How do you know? Okay, because you've got some nucleus and some organelles. And there's all these kind of specialized regions within the cytoplasm of the cell. And that's one of the things that we're going to do as we take a tour of the cell, is kind of hit the highlights. And one of the things that I want you to really think about as you study this material and as you prepare for an exam is word association. For a particular organelle, what is its major function? Okay, what kind of cellular processes are associated with that organelle? And then is there anything structurally that's special about that organelle that relates to its particular function? So that structure and function type of relationship. Images like this are a little bit dangerous, I think, for us in terms of our understanding of the cell. One of the real limitations is that these images are two-dimensional, and they really don't give you an idea of the true three-dimensional nature of a cell. Another thing that they're pretty dangerous about is that they give you this idea of there is a typical type of cell. And from the images I showed you earlier of the muscle cells or the nerve cells or the epithelial intestinal cells, a lot of cells look different from one another. And so this should be thought of not necessarily as kind of a typical cell, but kind of a composite of all the kind of things a cell might have. But in a particular situation, are there always 3 mitochondria in a cell? No. Are there always, this amount of endoplasmic reticulum? No. That the structure and function of a cell is the result of the fact that there are different kinds and numbers of organelles in different cell types. What would a muscle cell be loaded up with? A lot of mitochondria. Why? ATP production, and you need ATP for muscle contraction, for those filaments that make up the cytoskeleton to slide past one another, which results in the muscle cell contracting. Another thing I don't like about this particular rendition of a cell is it portrays the cell as being static. Okay? Here's where the nucleus is. Here's where the mitochondria are. And the cell is anything but static. It is a very dynamic structure. And there's a lot of interaction that takes place between the cell organelles. Some cell organelles are dividing and replicating. Some are moving from one location in the cell to another. There's a tremendous amount of intracellular chaos. And these static images don't really do a very good job of portraying that. In addition to microscopy, there's another important technology that's important in the study of cells. What if I were particularly interested in learning more about mitochondrial structure and function? How could I go in there and maybe pull out the mitochondria so I could study that cell organelle without the complication of the nucleus and the endoplasmic reticulum and the ribosomes and all that other stuff? Well, there are 2 technologies that are very helpful in that regard. And one of them is cell homogenization. Basically, that's just busting open a cell and letting everything leak out. And there's a lot of different ways you can do that. One of the more annoying labs we used to do, at least a long time ago, was we would homogenize plant tissues to study the chloroplasts. And we did that by throwing some sand into a mortar and pestle, a little bit of buffer, and spinach leaves, and ground them. Horrible sound, grinding sand. But you can homogenize things under pressure. You can use a blender to break things up. The other major technique is a technique called differential centrifugation. Y'all know what a centrifuge is? It's a device that you can put tubes in, and then if you put materials in those tubes and spin the centrifuge around, it subjects those tubes to increasing centrifugal force, and things start to move through the liquid in the tube at different rates. What's going to determine what moves where? Basically size or mass. Okay, so heavy things would be spun down easier than lighter things. So we can use these two particular techniques of homogenization, busting things up. Okay, here's intact tissue. Throw it in a blender. We've got a homogenized mess now. And then putting that in a centrifuge tube and spinning it and separating things based upon their densities. Okay? So this might be our original homogenate. And we spin this at 100 times G. G refers to the force of gravity. For 10 minutes. And what happens is the heavier things, like the nuclei, various pieces of broken cells and things, tend to be spun down to the bottom of the tube, and lighter things remain suspended. This is called the pellet, and this liquid is called the supernatant. What if we want to study something that's in the supernatant? We carefully pour it off into another tube, leaving the pellet behind, and then spin it faster, 20,000 G. And we get a new pellet. That pellet might be very enriched in mitochondria or chloroplasts, because they're pretty big. The supernatant might contain other smaller things like endoplasmic reticulum or ribosomes. So if we're a mitochondrial person, we might stop here and work with this particular fraction. If we're a person looking for cell membranes or ribosomes, we have to keep spinning at increasingly high speeds, pouring off the supernatant into new tubes and separating the organelles from one another based upon their differences in density. And there are actually some enzymatic tests you can do, because many times organelles have certain enzymes associated with them that serve as kind of markers to make sure that these really are mitochondria. So you can do an enzyme assay for mitochondrial enzymes, or you can do an enzyme assay for, microsomal membranes. Dr. Morton and I both did this kind of work in our PhD work eons ago. So very important tools, very important technologies. Okay, if I can get this to work, I'd like to show you a video that you may have seen before, you may not have. This is an animation. It's called The Inner Life of the Cell. And what it really tries to do is to give you some insight into what cell structure is really like from kind of a voyage inside the cell. What you're going to be looking at is a process that actually has three different names, and they all kind of mean the same thing. This process is called leukocyte extravasation, or transmigration, or sometimes referred to as diapenesis. So what kind of cells are we going to be looking at? What are leukocytes? They're white blood cells. And what is the purpose of white blood cells in your body? To fight off infection. Okay? And does anybody know how they do that? Yeah, they do a process called phagocytosis where they basically eat the other cell, like a bacterium. You ever seen an amoeba before? Move its pseudopodia and surround another cell and phagocytize it. So they crawl through your body, basically, and look for things that don't belong there and destroy them. And some of these white blood cells can signal other white blood cells, so you can start getting an immune response involving antibodies being produced as well. It's pretty complicated. Does anybody know where blood cells are produced in general? Produced in the bone marrow. Okay? You've got a bunch of cells in your bone marrow called stem cells, and they're constantly cranking out new red blood cells, new white blood cells, and new platelets. So initially, they're all released into the circulatory system. But a lot of the work by a white blood cell is done actually in the tissues. So if you get a splinter, for example, and it's got some bacteria on it, your white blood cells are going to have to move out of the circulatory system where they were released and into the surrounding tissues where they can encounter and kill the infectious agents. This process of moving out of the circulatory system and into the surrounding tissues is called leukocyte extravasation or transmigration or diapetesis. In order for that to happen, the white blood cells are moving along through the blood. They've got to grab onto the wall of the little vessel that they're traveling through. And then once they've grabbed onto the wall, they've got to squeeze between the cellular junctions that join the cells of that vessel. They've got to kind of crawl through the junctions of the vessel so they can get out into the tissues. That requires a real change in the shape of the cell. And those shape changes require a tremendous amount of activity within the cytoplasm to make those happen. So this inner life of the cell is an animation done by Harvard. It's probably maybe 10, 15 years old now.
At least 10.
Yeah. But it's still very good in terms of illustrating the dynamic nature of the cell, the three-dimensional nature of the cell, and the complexity of the cell. Some of the stuff that you see in the images will look familiar, other stuff won't. What I'll try to do, if we have time, is once we've gone through the tour of the cell, we'll come back and show this again and point out some of the stuff that you might not have recognized before. So let's see if my internet connection is going to cooperate here. Now there's just some music in the background. I'm not getting the music.
You have the sound off on your computer?
I don't think so.
I think the sound is what makes it interesting.
Pardon me?
Sound makes it interesting.
Yeah, I know. The sound is... I think that's my favorite image of them all.
Is the amplifier on?
Yeah, good question. the white blood cell. One of the things that I hope you can come to appreciate in the course of this class is that these cell parts that we're going to spend a fair amount of time talking about are just organic molecules. Okay? They're proteins, they're carbohydrates, they're lipids, and they're nucleic acids, all interacting with one another. So what that video was showing you was some of the molecules and some of the things that were happening during these various interactions. So we're going to be pulling those apart and looking at a little bit more detail throughout the course of the semester, various cellular processes within the context of a particular phenomena like cellular respiration. So Thursday we will continue. We'll talk a little bit about prokaryotic, eukaryotic differences, and then dive into the various cell organelles, trying to give you a relationship between structure and function of those particular cell organelles. And so, if you can, I would urge you to do reading ahead in Chapter 4, because if you kind of know what's coming, it's easier to figure out where we're going. So, that's probably a good place to break.